Hi dear children, today we are going to study about respiration in plants. Respiration in plants, the first step is glycolysis. Glycolysis starts from the cytoplasm. It is actually the breakdown of glucose and this is the first step. Let us see what is happening there. Actually, glycolysis is actually a 10 series of steps. Chain reaction is happening and all the steps are enzyme controlled. Different enzymes are there and each steps ATPs are used and even ATPs are produced and even NADH2 is also produced. Before starting this, let me tell a general fact. The carbohydrate which is produced during the time of photosynthesis is actually sucrose. Actually, this sucrose get converted into glucose and fructose by invertase enzyme. And this glucose and fructose undergo respiration from cytoplasm. So, this glucose and fructose is actually undergoing respiration. Normally, we used to say glucose is the end product of photosynthesis, but it is not like that. Sucrose is actually the end product, but later it is converted into glucose and fructose. This both can undergo respiration. Okay, now let us see it with glucose. Okay, children, the first step is glucose is being converted into glucose 6-phosphate. It is in the presence of an ATP. Why? Because ATP will give one phosphate so that glucose can convert into glucose 6-phosphate. This is happening in the presence of an enzyme called hexokinase. See, now what is 6 meaning? 6, the meaning of 6 is not that 6-phosphate is there. It is not like that. Glucose actually contains 6 carbon. Into that, in the 6th position, phosphate is added. That is why the number is coming. Anyway, you might have studied in chemistry this representation. Okay, glucose 6-phosphate means over the 6th carbon, phosphate group is attached. And that phosphate group came from where? From the ATP. As a result, ATP will be turned to ADP. So, in the first step, ATP is used. Now, when this comes, like this steps comes, you should be able to understand what is a change. Okay. Okay, now next step. Now, next step is just a rearrangement in glucose 6-phosphate. They are just turned into fructose 6-phosphate. It's because this glucose and fructose are the same. They are just like the isomeric forms. Both are the same. So, both can be interchanged. So, simply glucose 6-phosphate is converted into fructose 6-phosphate with the help of an enzyme called isomerase, since it is an isomerization reaction. Next step is, see the change, glucose 6-phosphate is converted into glucose 1,6-biphosphate. So, what is added? Biphosphate means one more phosphate is added. Who will add this phosphate? Obviously, that is the ATP. ATP will give one phosphate again. So, another ATP again comes and they turn to ADP. So that again a phosphate group is added to which carbon in the fructose? First carbon, right? Already in the 6th carbon one phosphate group was there. Now into the first carbon position again one more phosphate is added. So it came to fructose 1,6-biphosphate. Okay? Okay. Now till now there are 2 ATPs used. Now we reached up to 1, 2, 3 third step, right? Now what is the 4th step? Fructose 1,6-biphosphate. Now it is splitted into 2. It is splitted into 2 forms, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So 6 carbon fructose is converted into glyceraldehyde 3-phosphate, which is of a 3 carbon form, and dihydroxyacetone phosphate, which is also a 3 carbon form. So this is the 4th step, splitting up of fructose 1,6-biphosphate. And one more thing, children. This splitting is being actually catalyzed by an enzyme called aldolase. All steps are catalyzed by certain enzymes. What's the next step? The 5th, as I told, this glyceraldehyde 3-phosphate and dihydroxyacetone phosphates are the similar forms. They are the isomeric forms. So, in this step, the dihydroxyacetone phosphate is being converted into glyceraldehyde 3-phosphate. It is an easy step because they are the isomeric forms and this reaction is catalyzed by the enzyme. Which will be that enzyme? Isomerase enzyme, a particular isomerase enzyme. If this is a process, now for the further reactions, how many glyceraldehyde 3-phosphate will be there? 1 or 2? Yes, it will be 2 molecules because already glyceraldehyde 3-phosphate has been formed from fructose 1,6-biphosphate. Again, this dihydroxyacetone was phosphate was converted into glyceraldehyde 3-phosphate. So there will be now 2 molecules of glyceraldehyde 3-phosphate. Now all further steps have been done with this. Now here what is happening, we can see in 2 molecules of glyceraldehyde 3-phosphate what will happen. Okay, in order to make it easy, I have told it separately. I have it is drawn separately, so it is easy for you to understand. Okay children, in this step what is happening, glyceraldehyde 3-phosphate, that is 2 molecules of glyceraldehyde 3-phosphate is being going to convert into 1,3-biphosphoglyceric acid. Keep in mind, glyceraldehyde have to convert into glyceric acid. Okay. Now here actually the reaction is oxidation reaction. We know oxidation means removal of hydrogen and electron. So what is the meaning? From glyceraldehyde 3-phosphate, when hydrogen is removed, they will be turned to phosphoglyceric acid. That is why they are undergoing oxidation reaction. But here a slight change is there. Glyceraldehyde 3-phosphate is converted into not simply phosphoglyceric acid, but they are converted into biphosphoglyceric acid. See children, this is the general formula of glyceraldehyde 3-phosphate. You can see hydrogen ion from carbon number 1 is removed, and that is been taken by NAD and converted into NADH2. Then again a phosphate group extra is added. As a result, you can see 2 phosphate group in carbon number 1 and carbon 3. See the equation formula, you will understand. In this way, during this reaction, we can say NADH2 is formed, right? The hydrogen ion are accepted by NAD, and as a result, what is formed? NADH2 there. Okay. Okay, next step is very easy children. Here 1,3-biphosphoglyceric acid is converted into 3-phosphoglyceric acid. So what is the change? The already added phosphate to the first carbon is removed in the next step. That is removed again. So from the first carbon position, phosphate is going out. As a result, it is turned into 3-phosphoglyceric acid. That is a removal of added phosphate group. Now who will take this phosphate group? The removed phosphate group will be accepted by our ADP energy unit, which will be there in the cytoplasm. As a result, ATP will be formed. So this is the first step in glycolysis where ATP is synthesized. In the first and third step, which we have already seen, there ATP was used. This is the first time where ATP is synthesized. Okay. Okay, next step is very easy children. Here there is a rearrangement of the position of phosphate group. Already 3-phosphoglyceric acid means phosphate group is there in the third carbon. In this step, here the phosphate group is changing their position to the second carbon. Okay children, see the picture. This is the first picture is the this is phosphoglyceric acid, 3-phosphoglyceric acid formula. There you can see phosphate in the third carbon. Now there is a rearrangement of phosphate group to the second carbon, shifting to the second carbon. So again we will not call them as 3-phosphoglyceric acid. Instead we will call them as 2-phosphoglyceric acid. In this way 2-phosphoglyceric acid is formed. In how much 2-phosphoglyceric acids will be there? 2 molecules, right? Everything is 2 molecules already we have told. Now next step is this 2 molecules of 2-phosphoglyceric acid will remove 1 water molecules in the next step, and as a result it will be converted into phosphoenol pyruvate. When a water molecule is removed from 2-phosphoglyceric acid, they can be converted into PEP, that is phosphoenol pyruvate. That is the next step, removal of water molecule. Okay. Now the last step, PEP is being converted into pyruvic acid by removing a phosphate group. When a phosphate group is removed from PEP, that is phosphoenol pyruvate, what will be formed? Pyruvic acid or pyruvate. So this is the last step of glycolysis. 2 molecules of pyruvic acid will be formed when a glucose undergo glycolysis. So these are the all steps. There are many enzymes taking part and many ATPs, NADPH2s are formed. Finally, you can see children, from 1 molecule of glucose, 2 pyruvic acid will be formed, and where is the ATP is formed and where is the NADH2 is formed. How much ATP is formed? How much NADH 2 is formed and how much ATP is used, in which step this has been done. Make it very clear and thorough, because it is, and for exam it is very must to know the positions where this process is held. Okay. So thank you children for watching my video. Hope it was useful for you. If you like my video, please subscribe my channel Mithul's Biovlog, and please press the bell button so you will get easy notification of my further classes. Thank you. Have a nice day.